All right, so for our project, we chose to focus on the Brain Initiative, and the goal of the Brain Initiative is to map the brain and get a more thorough understanding of how the brain works on the nanoscale. We specifically chose to focus on the neurological disorder Creutzfeldt-Jakob Disease.
So Creutzfeldt-Jakob Disease is a rare, rapidly progressing brain degenerative disease. And we know that it's caused by prion protein mutations, but we really don't know a lot about these prion proteins themselves. So, for example, we don't know a lot about their exact structure or their exact purpose. So we hope to design an experiment that would use different methods of microscopy to do a side-by-side comparison of healthy prion proteins and infected prion proteins. So this would help us to gain a better understanding of the prion, which is essentially our goal, because this would lead to better methods of diagnosis in the future. And it would also address the Brain Initiative because it would help us to better map the brain in more detail. So what exactly are we trying to answer? So our fundamental question is, what are the fundamental differences between the cell membranes and nuclei of healthy prion protein cells and infected prion protein cells? So in this image, you can see that this is a really simplistic model of the prion protein cell. That's because scientists really don't know much about it right now, and that really isn't helpful when you're trying to solve something like Creutzfeldt-Jakob disease. So answering this question would really help us to gain a more complete understanding of CJD, and it would also help scientists to develop more effective methods and would lead to improved treatment later on.
Okay. Creutzfeldt-Jakob disease is a rare, degenerative, rapidly progressing, and invariably fatal brain disorder discovered in the 1920s by German neuroscientists Hans Gerhard Creutzfeldt and Alphonse Maria Jacob. This disease is caused by misfolded prion proteins building up in the brain, and these mutated prions infect other healthy prion proteins, eventually leading to the abnormal prion proteins destroying their neurons in the brain, thus leading to a wide array of symptoms. In the early stages of CJD, symptoms include failing memory, blurred vision, behavioral changes, and lack of coordination. As these symptoms are similar to other more common brain disorders such as Alzheimer's, some patients with CJD can be wrongly diagnosed. But the later stages of CJD have more extreme symptoms such as dementia, involuntary movements, blindness, and weaknesses of extremities.
All right, so prion proteins, a mutation in them is what causes this disease, but scientists don't actually know the function of prions when they're healthy and functioning normally. So what they do know is that they're highly expressed in neurons and that they play some sort of regulatory role in the nervous system, though they're not exactly sure what it is. In a study done with mice where they had nullified the prion gene so it could not perform any function and just wasn't working properly, they noticed that the mice were more prone to oxidative stress and unnecessary apoptosis, which shows that, like, once again, they are necessary because they lead to a decreased health if they're not there, but they don't know why that is. They also performed a study with zebrafish embryos where they used green fluorescent protein to tag the prions, and they noticed that the prions tended to accumulate in places where the cells were in contact with each other, so that could mean that they may play a role in cell-to-cell communication. And also, in another study done with zebrafish, they nullified one type of prion protein, and in this stage, the zebrafish embryos did not make it through the gastrulation period. And when they tried it with a different prion protein, they made it to early larval stages, but most of these zebrafish had serious morphological deformities. Oh, and these pictures are both trying to show how the protein changes from normal to when it's mutated, but if you notice, they're not exactly the same picture because scientists don't know for sure the structure and how it works.
So the potential impact. So there are some pretty significant potential impacts. One is, of course, a better way of diagnosing CJD as well as other prion diseases. So it could also accelerate advancements in the research of other prion diseases like Alzheimer's, for example, and it would bring us one step closer to a cure of all of these diseases.
Currently, to diagnose CJD patients, doctors perform numerous tests such as magnetic resonance imaging, electroencephalography, neurological and physical examinations, along with cerebrospinal fluid analysis, which is extremely painful. And EEG is the measurement of electrical activity in different parts of the brain and the creation of a visual trace of that activity. And the cerebrospinal fluid is produced by choroid plexus in the brain and is absorbed into the bloodstream to deliver nutrients to the brain.
So the types of microscopes we're going to use in our design are the cryo-electron microscope. And so this one is a cryo electron transmission microscope, which means that first the sample is going to be frozen before it's put into the microscope to create contrast, and then electron beams are going to pass through the sample, and the microscope is going to read them and then create an image from that. The second kind we're using is atomic force probe microscopy. And in that one, a needle probe is ran along the surface of the sample, so it actually comes into contact with the sample and measures its elasticity by touch to create an image. And these, the one farthest to your right is the electron, and the one closer is the atomic force probe. So for our plan to address this issue, we're going to take spinal taps to attain spinal fluid from patients with CJD and some willing participants who are willing to subject themselves to getting a spinal tap, which, yeah. And so we're going to perform the cryo-electron microscopy, and we're going to look for any changes in the structure of DNA. So specifically, we're going to see if the DNA changes locations within the nucleus or if the nucleus has any significant changes in size or shape. And in the atomic force probe microscope, we're going to analyze the surface texture of the prions and see if maybe there's any protrusions or indentations particular to the CJD ones that are not in the healthy ones. And hopefully, if we understand the structure of those, we can understand the structure of the cell membrane of cells infected with it. And these, on the top is an electron microscope, and on the bottom is an atomic force probe microscope.
So there are a few challenges with this approach. The major challenge stems from the fact that CJD is really rare. So this would mean that it would be difficult to find patients who would be willing to subject themselves to spinal taps, of course. And then also it would be really difficult to find definitive patterns because we would have a really limited amount of data with limited patients. All right.
That's all. These are our references.
Oh, and also CJD kills only less than 1,000 people in the U.S., so it's kind of a small number in comparison to the population of the U.S. Other prion protein diseases include Alzheimer's, like the other group talked about, and Kuru, which is found in Papua New Guinea, that's spread through cannibalism, and it's infectious.
So you mentioned the challenges of getting samples from humans. So what would be the alternative to using human samples?
Yeah, if we couldn't use humans, we would probably use zebrafish because they're easy to look at under a microscope, and they have similar DNA to humans.
Is there a mouse model?
There's been trials done on mice using it, so that's also an alternative you could use. I would say either one of those.
How come you don't use like other primates?
Just because in testing it's easier to use smaller animals and more like cheaper, readily accessible animals. I mean, and it's like, I don't know, you run into more ethical issues when you're using larger animals for testing. So.
There's also another factor, which is that Creutzfeldt-Jakob develops over a very, very long period of time. And this is also one of the challenges. It takes about like 30 years before you develop the symptoms. So if you use an animal model that has a lifespan that's much shorter, you can expect to see the symptoms much faster.
That's why fruit flies are used a lot. Kind of. Yeah. Well, you increase it to like 6 figures, I'll do a spinal tap.
You got it. Done and done.
So, I have a question. So, why did you think about looking at the location of the DNA? What would be the effect of different locations?
Well, because we know so little about it, DNA is just like the best starting point we could think of, because if we notice like a difference in structure, it could maybe be due to figuring out what kind of mutation occurred in the DNA, or like what kind of secondary structure changed in the protein, and that would be a good basis to start further research on, and possibly develop a more accurate way to diagnose people so they don't have to go through such extensive tests.
Yeah, I can't.
Yeah, can you say that louder?
Are prions completely resistant to chaperone repair?
Yeah, well, they are, because like you said, they accumulate for years and years. It's called an incubation period, and it just, I mean, they can't be stopped, and they don't contribute symptoms. So, for some reason, yeah, they go unchecked.
Only Kuru was the infectious disease. This one's like sporadic. Mostly 85% is sporadic CJD, and the rest is like familial.
Yeah, you can get it, but it's usually the only way to get it between people is by like having a tissue, like if you were getting an organ from someone or some sort of tissue-to-tissue contact.
Yeah, or if you were eating like bad meat, you get it too.
So in the case of Kuru, since you mentioned it, it's not really contagious. I mean, the only way the pygmy of New Guinea, which has been reported, got the Kurus because there is a ritual out there that is to eat the brain of the person that passed away, right? And it turned out that there's a high prevalence of Creutzfeldt-Jakobs in that population, and this is how the disease has been passed on, actually. So it's not really infectious in the common sense. It's very unique.
Isn't that kind of like the way mad cow disease is done?
Yeah, it's just,
Yeah.
it's the same thing,
Oh, it's the same thing.
essentially.
Oh, okay.
If there's one more question, then one more
I
question.
have one quick question. You mentioned earlier it said it was rapidly, that it spread kind of rapidly once you have it, but the incubation is slow.
Yeah, the incubation is slow because as the protein mutates, it can affect other proteins, but it takes a while for that to happen. The symptoms don't usually display themselves till like a year before. Once you're diagnosed, you usually have less than a year to live. It's pretty bad. Like you don't really know until you get symptoms, and then it gets really bad really fast.
Right.
Oh,
I
there's an-
think we, is it a quick question?
It's a quick one.
All right.
About 2 weeks ago, I was listening to NPR in the morning, and there is an ongoing study right now. A woman had the disease, and they were talking about genetic differences. So it's kind of like an up-and-coming big topic in science because of the ramifications for other brain diseases. So.
Oh yeah, because like prion, it's a type of prion disease which covers a lot of different things, and it's just like a step forward in understanding like dementia and deterioration of the brain. So research could be like applied or relate to other diseases as well.
All right. Thank you, guys.